I'm going to tell you a little secret, which is the title in my talk is a title I give to everyone for a talk, and then usually the night before I give the talk, I really sit down and think about what it is that I want to talk about. And I thought it might be really fun in the context of a lot of the awesome talks that we've heard about already this week, particularly talks from Nancy and Lisa and the workshop that Bogdan is going to lead tomorrow, to really focus on some of the questions about what's going to happen in the next 5 and 10 years when we start to center genomic information more and more routinely, both in clinical care and also in realms of public health and society. So I'm going to ask you to bear with me because I really just sort of put this together in half research talk, half a little bit thought experiment. So I hope you'll also, it'll be more of a dialogue. And I should start and just say my disclosures. But I hope that by the end of the 40 minutes, we'll really start to think about some of these big questions that are going to face our field in the coming decade. You may walk away with this thinking that I've just spent 40 minutes doing a propaganda exercise on how cool and fun it is to work with biobank data. That actually might be true, and that's showing my own biases. But truly, all it is about working with data that's truly collected in an unselected way, and mainly, and a little bit agnostic. We got there sooner with genomic data, but health systems data has been there for a while, and other types of data are coming online. So you can think about this more generally too. But let's start with genomics. Okay. So today, who has their genome assayed? Most of genome assay, whether it's array data that you're generating or sequence data through clinic or through research, this is happening mainly in health systems and actually increasingly in health systems. Previously it was happening mainly in research. I think it's really balancing out now, and toward the future I think we're going to see health systems generating more and more of the genomic data that we have around the world. The people who get sequenced are either patients with suspected genetic disorders or relatives of those patients sometimes. People who volunteer for research, so volunteer for cohort studies or biobanks or clinical trials, or consumers, people with their credit card who are interested and have funds to go and purchase genetic data about themselves, or at a doctor's office can pay out of pocket for tests that aren't covered. That's today. But the question is what's going to happen in the future, and we all know well in this room that we are studying a technology that's dropping precipitously in price. Well, it's kind of stalled in the last 5 years, but it's still relatively cheap in health systems these days compared to any other type of test that health systems routinely run. But the question is, will it really be that because of the cheapness of technology and all the wealth of information that it contains, in the future everybody will be sequenced? And many people would postulate that the future will look something like this. It will be truly an era of genome-first medicine. In other words, we sequence your genome and we can infinitely look up and test that genome over the course of your life to find out information which we think is going to be important to maintain your health through the course of your lifetime. And this is a promise really that came 22 years ago with the enormous achievement of sequencing the first human genome. I think at the time many people thought we would already be there yet, but 2 decades later, I think we're still really on the precipice of being in this era, and some people are doing it, and I'll talk to you a little bit about what that really looks like. The potential is huge. So the potential is that we will have increasingly sophisticated molecular diagnostics that help us achieve more accurate diagnosis, and sooner if you start to look at genomes first. We can also use genomic data not just for diagnosing disease, but also in healthy people or newborns to predict risk throughout people's lifetime, and this is what you just heard Iran speak about. Another aspect where we can really think about leveraging this information, which is one that I think a lot about, is really at the population level. So not just the individual level, but also at the population level, and how we think about resources and where those resources would be most effective for using genomic information for improving population health. But as we're thinking about those potential benefits, we have to really address the challenges ahead, and this is one of them. I think this slide, this figure, which is an updated 2022 version of a figure that came out in 2018 from a paper by Alicia Martin, is now very famous because you see it in almost any talk in genetics. And it's really that genomics is failing on diversity. I don't think I have to tell this room the reason why is that we have, for the last 20 years, spent most of our time sequencing people of Northwestern European ancestry, and many other humans around the planet are not represented in these massive databases that we built, or underrepresented. And that turns out to be a problem, and it creates problems of generalizability and transferability of knowledge from these databases into tools and findings that help us understand the health of all humans around the world. And this is not just in basic research side, by the way, folks. This is also in clinical research side. It's all the way through the pipeline of bringing basic research findings into clinical care. There's a lack of diversity in clinical trials. There are a lack of resources in low and middle resourced areas in the world, or areas of the US. And there are many regions and populations that are just not included in the type of biomedical research that we do. And one of the reasons for excitement about biobanks is because they really can be translational engines for genomic research. Here's a figure from a paper, from a review actually, that Noor Abul Hassan and I wrote a couple of years ago. And this was generated by contacting our friends who run biobanks around the world and asking them some questions about the biobanks there, and then sort of constructing this figure which looks at somewhat the history of biobanks, where in the world they've arisen in the last 2.5 decades, and then also on the y-axis the proportion of non-European ancestry participants in the biobank. That's a measure of diversity. It's not the only measure of diversity. We should care about diversity in multiple dimensions, but just for fun, this is one of them. And you can see that, of course, if you have biobanks that have come up in other areas of the world, in other continents, that's going to increase diversity. But most biobanks are in these green and blue, so they're in Europe and North America. Good news is over time, and with increasing awareness of this problem and concerted effort to include diverse participants in biobanks, we do see that diversity is increasing. And it tends to be this way for biobanks a little bit more compared to cohort study, by virtue of the fact that biobanks tend to be, not always, but tend to be a little bit more population unselected, all comers, whomever walks into a health system. So in that way, you sort of can enrich for diversity. So I've been a faculty at the Mount Sinai Health System now for coming up on 10 years. November is my 10th anniversary. And the reason that I went there to start my faculty career was because they were one of the institutes back in the early aughties that actually had a biobank endeavor. And subsequently, unlike other biobanks here, our biobank hasn't grown so much in size. Today we have about 70,000 participants, and if you know biobanks, that's sort of small scale compared to some of the really huge biobanks that are out there. And and we all know sample size, particularly for common diseases, is king in terms of being able to have power to identify what's going on in genetic architecture. However, one of the areas where this biobank shines a lot is that it is highly diverse by dint of being situated in New York City. So a little bit about Mount Sinai. Mount Sinai is a healthcare continuum, about 8 hospitals, hundreds of ambulatory care clinics all over the 5 boroughs of New York, feeding into one epic system. That's our data warehouse. And it's situated in New York, which is an extremely diverse place, historical point of entry from all over the world for hundreds of years to the States. And that's very much reflected in the citizens of New York, and we're very proud of our diversity, and it's usually what we're known for. Such that if you look at the participants of Biobank broken down by race and ethnicity, you see that 65% of participants are non-European ancestry, with big representation in both Hispanic, Latino, and African American communities in New York City. And all of this data is linked not only to electronic health information that's in the record and digitized, and increasingly these days that's starting to become very dense and rich data, not just ICD-9 billing codes, but digitized image data, digitized histology data, pathology data, lots of data. Also survey data that we collect when people volunteer to the biobank. And we've generated, both through research grants and industry partnerships, genomic data for almost all the participants. So with this very diverse biobank, you can start to go in and untangle some of the questions about what is driving health disparity in genomic medicine. And maybe my talk is a nice segue from Nancy's talk the other day. So I'm just going to now paint the picture of what that looks like in a health system in New York City. So the first thing to point out is, and I should say all this work that I'm going to show next is from a paper that just came out last summer, led by Gillian Belbin, who is a wonderful grad student, then postdoc, then faculty in my institute, now working at GenCove and doing some really awesome work there. But she started to look at, just to ask a basic question, what does your health system think about you in terms of race and ethnicity? And it turns out that health systems generally do a poor-ish job of that. And here's looking at data over a period of 7 years for 30,000 people in the biobank, where we have survey data, which we can treat as ground truth for this experiment, and counting 1.3 million visits into the health system. So each individual comes into the health system, variable amount of visits, sometimes hundreds, over the course of that time. And you can see for every person, there's going to be a flip-flop in terms of what is recorded in the health system about your race and ethnicity. For no individual, 100% of the time, does the health system agree with how people report in the survey. But it's also different across populations, where we do much, much worse for Hispanic, Latino, and Asian populations in our health system, abysmally for Native American or people who would identify as multiple ancestries in our health system. But of course, race and ethnicity is only the tip of the iceberg, particularly in New York City. And if you ask another question, where were you born, where were your parents born, where were your grandparents born, then you start to get a much more nuanced and broader understanding of diversity and self-identity. For example, 65% of participants in the biobank have 3 or more grandparents born outside of mainland US. And this is from up to 160 countries around the world. So that starts to give you now a more nuanced sense of the type of diversity, which itself is linked to people's cultural, environmental, social experience. And we can see some of that nuance and complexity still now just looking at self-reported or EHR captured data, and not yet looking at any genetic data. When we start to look at the bottom row at the type of population categories we typically use in research, like European ancestry, African American or African, Hispanic, Latino, South Asian, and the type of regions of the world where people in the biobank have recent ancestry. And you can see for many of these categories, they don't line up neatly. And really, often this is masked in research because we don't even ask those questions. We don't ask questions about country of origin of your grandparents often. So this is just giving you a snapshot. Now, of course, we know genetic information can start to help us unfurl this level of diversity. And here's a tool we use called principal component analysis. I think most people in this room are going to know what that is. And I'm going to point out to you a few things about this principal component analysis, which is of Biomery participants in New York. One is, I've painted on here, reference populations from 7 continents around the world, and you can see that the grey dots, which are Biomery participants or New Yorkers, overlap all 7, which is not surprising given the picture I just showed you, given people's recent origin from all around the world. So the participants in Biomery, rather than really representing discrete race or ethnicity populations, are rather representing a continuum of genetic diversity that's kind of a snapshot of the world. It's not everywhere in the world, but many places in the world. In my lab for many, many years now, we have been very interested in using genetic information to help us understand nuances of fine-grained population structure. We are particularly interested in the population structure that's occurred in the last 5,000 years. Why? Because we think that's a very interesting time in human history, both because we've undergone an enormous Log 4, Log 5 exponential growth as our species. Also, there's been an enormous amount of intercontinental travel in very complex patterns. So we try and build tools and methods to try and disentangle that. One of them is using haplotypes shared identical by descent in recent ancestral time to reconstruct local patterns of genetic similarity and dissimilarity. And I'll just shout out, I won't tell you about it, but I'll just give a shout out to some of the tools that we've built, particularly most recently a tool called Eyelash, built by Ruhi Shimarani, who is here in the audience, who will hate me pointing him out. Oh, there he is, red shirt at the back. And this is a great tool because it uses local sensitivity. So I should hands up, and Sasha Gusev, I know, was here in the first week talking, who built one of the tools we had previously used, a standard in the field called Germline, but I was there when he built it. That was 15 years ago. So we're using more modern hashing techniques in computer science. I say we, this is really Ruhi's work. And then 6 months of optimization to actually get a tool that really speeds up the process and allows us to detect IBD in databases of 100, 500,000, a million people, where you're detecting billions and billions of pairwise IBD. But this is published work, and if you're interested in that, the paper's on the slide. But back to Biomery. Oh, did I knock something off? Can you still hear me? Yeah? Okay. Did I do that? Hello? Okay, sorry, goblins in the system. Okay, so back to Biomery. When we apply these tools to detect this more recent population structure in New York City, and then we use unsupervised methods to sort of learn the topology of that sparse matrix of pairwise sharing. Then we start to find clouds of people who are more likely to share IBD within the cloud than outside the cloud. We call these IBD communities. We can overlay information that we've collected about race, ethnicity, or country of origin, and most of the time these clouds are highly predictive of country of origin information. Some of the times they're not. Some of the times it's because they're more predictive of cultural information, like whether or not somebody's Ashkenazi Jewish, whether or not somebody's Garifuna. And some of the times we don't get good predictive labels. We're not sure why. We can guess, and it's a fun thing to do, but we'll be here all night. Okay. So that tells you a little bit about population structure and the type of population structure that's actually going on in a health system using genetic tools to disentangle it. Now let's start to think about what this means for bringing genomic information into healthcare. Let's start with monogenic risk, because that's the simplest type of information and it's the most common information today in health systems. And I don't have to tell you what monogenic risk is, but I'm going to start with a very, very simple example. I'm going to start with the BRCA1, BRCA2 genes, which we've known about since 94, 95. So for a long, long time, these are probably some of the most well-characterized genes in the human genome in terms of clinical impact. And screened for. So very many people who are at risk or showing signs and symptoms of hereditary breast and ovarian cancer syndrome get access to genetic screening tests. Often their relatives do too. So we do a lot of screening. It's a success story of how we bring genomic research into clinical care over the course of the last 30 years. Nonetheless, even with all that evidence behind it, if you go into the Mount Sinai health system, 74% of people who are walking around with a pathogenic variant of BRCA1 or BRCA2 are unaware of that. And most people don't know they are harboring that pathogenic variant until after they are diagnosed with an associated cancer. Moreover, even though people who harbor a pathogenic variant in those genes are at higher risk of many cancers, it turns out that the prevalence of that risk varies quite considerably across populations, and many of whom we've really very much understudied for BRCA risk. So, for example, in New York, across the 17 different communities that we're able to pull out, we see a tenfold difference in prevalence risk across populations. The highest, probably unsurprising to some of you in the room, is in Ashkenazi Jewish populations in New York. The reason there is because there's 3 canonical founder variants at appreciable frequency segregating in Ashkenazi populations that we've known about for a long time. Lisa, you have your hand up. Yeah, so that's a great point. I have another slide just coming, and I'll come back to that point. That's a great point. You've actually beaten me to the punchline. No, it's great. It's great because it very much relates. So we see the highest rates in Ashkenazi Jewish populations. We see much lower rates, for example, 1 in 500 risk in some Caribbean Hispanic Latino populations. But despite the fact that we can show using health systems data that everyone who harbors a pathogenic variant in this gene has very similar risk, about 25% risk in their lifetime of having a personal or family history of one of the associated cancers. If you are Ashkenazi Jewish in our health system, you are twice as likely to have had a genetic test and have been screened than other populations, which I think might be getting to your question, Lisa, which is, we don't know why. This is just at this point observational, but suspect this may have something to do with either population-based or provider-based knowledge of risk and therefore change in health management and recommendations. And my phone has just turned on. I don't know why. When it happened to us, it changed our lives forever. Okay, bear with me. There we go. Thank you. Some gremlins at work today, I don't know why. Or other more complex reasons about access and who is accessing screening and who is not accessing screening. Yeah, I know. I did some Facebook ads. It's gone. So there are a whole complex set of reasons, and I certainly not with this data, could point a finger at any one, but we can start to see what's happening in the health system and then start to address why these things are happening. Another thing that's happening within health systems that I think a number of groups are starting to publish on and pay attention to, and we have been too, is that whether or not, which population you are from or identify as can matter in terms of how well genetic testing performs for you. And in this case, we're asking how many times a test comes back with uncertain, conflicting evidence for pathogenicity, or VUS, as we call it, versus a positive or negative. So where you want to be is you want to know if it's a positive or negative. You don't want to be in the middle where we're not sure. And from the same study that we used for BRCA variants, it turns out that you are much more likely to get an uncertain finding from a genetic test at the BRCA genes if you are non-European versus if you are European. In another study that we are writing up now, hope to publish soon, we dug a little deeper into that. We looked not just at BRCA1 and BRCA2, we looked at the entire set of genes in ClinVar, ClinGen, and I looked at this in a more global scale and actually sort of brought genetic ancestry into it so we could start to implicate what was really going on. And it turns out it's really, in our analysis at least, the proportion of genetic ancestry in your genome, not necessarily how you self-identify or self-report, that is correlated with your increased chance of getting an uncertain result from a genetic test. Back to biobanks. So what can we do about this? It's one thing to point out a problem, it's quite another coming up with a solution. And this is where biobanks come back into play, because biobanks are not just massive troves of clinical data that we can repurpose for research. They're accelerated ways that we can take research findings and bring them into clinical settings. So here's an example that I'm going to talk to you about. And I like this example because it is the complete yin and yang of the academic experience. So we were interested in the early days when I first got to Mount Sinai, and Gillian Belbin was my first grad student. Her name is going to come up a good bit in this talk because a lot of this is her work. And we were very interested in leveraging biobanks to uncover rare monogenic diseases. And the reason we were interested in that was because we had built out all these, we thought, really nice methods to detect IBD, and IBD haplotypes are tagging rare variants. So we only had array data at the time, but we were able to use IBD to detect rare variants in the biobank. So we thought, why don't we use the medical phenome data to really explore if there's any evidence for association for disease at these rare variant sites. And because we were really thinking about rare variants and we knew there was a lot of founder effects in New York, we were really looking at recessive disease. So that's this experiment, and we set it up as a FEWAS. Immediately, what jumped out was a paper that we published back in 2015. Our finding we got 2013. We actually almost got it, I want to say it was almost day one, because this is a variant which in 2 copies breaks height. And that was magic, because height is such an easy human trait to work with. And when something makes humans on average 10 centimeters or 10 inches shorter, you notice it straight away. So we found that variant. Of course, it's doing something more serious in collagen, and it's linked to a collagen disorder. And we were able to cleanly identify that story using biobanks and publish it. But that was not the top hit in the FEWAS. The top hit was the FEWAS was something that was in recessive state associated with biliary cirrhosis as the top hit, and a few other livery type things underneath that. And that took us about 10 years to pin down. So this is what I'm saying about the yin and the yang. This was a much more complicated story, and I'm going to talk you through it, but I want to point out at the end of the day, we would not have been able to figure this out by ourselves. We really needed to reach out both to molecular biology colleagues and cell biologists, and reach out to clinical colleagues to really help elucidate what was going on here. So our initial finding was that 3 individuals who had these 6 haplotypes that were identical by descent all had an association to biliary cirrhosis. We first started with fine mapping. Fine mapping meant that we did whole genome sequencing across those 3 individuals, and then we looked at the common shared boundaries of the edges of the haplotypes that they shared, and then we looked for the variants that were in the sequence data, in the common shared boundaries, and there were hundreds. But we were able to use a couple of logic to help us narrow this down. We were able to throw out anything that was common, because if this is a rare disorder, we assume that the variants are rare. And we were able to also intersect this with ClinVar, because we still had hundreds at that point. So we used ClinVar to help us narrow it down even further. And we didn't have any sort of golden goose egg in terms of a coding variant, but we did have a variant that looked like a splice variant. It probably took us about 5 years to get the experiments right, but we were able to go in silico and show that if you knock out or modify this variant, it impacts splicing, and it knocks down exon 23 and 24 of this gene, ABCB4, and that results in a non-functional protein. We also had a little clue, not in the beginning, but kind of at the end of this odyssey when it popped up in ClinVar, that this specific variant was also linked by one submitter in one family to a childhood hepatobiliary disorder. But in our health systems, we had 5 individuals who harbored 2 copies of this splice variant. So we went to our clinical colleagues, and we had our clinical colleagues in liver disease with subject domain expertise in liver disease actually go into the health records of these participants. So first of all 5 had quite elevated serum liver enzymes and liver function tests. Even more worryingly, 4 out of 5 had a diagnosis of cirrhosis, and the 5th had liver steatosis. 2 had undergone liver transplant. 1 was found to have incidental hepatocellular carcinoma on explant. So all indications that each of these 5 individuals were experiencing severe liver disease. But this still wasn't a screaming finding, because if you look at the etiology of each of the 5 individuals, it was actually each quite distinct, ranging from different types of liver diseases, alcohol-induced cirrhosis, through primary biliary okay, this is why I didn't become a doctor, that one, and all the way to something that's called cryptic cirrhosis, which basically means in medical terminology, we don't know why your liver is sick. And so our best guess at this point is that there might be some mechanism upstream of bad liver disease that is being impacted by this particular variant, such that people go down a path for bad liver disease, but it can be distinct. We see them in their health system when they start to get really sick. The other very interesting thing about this variant is even though we were looking at it as a recessive variant, when you actually go back and you look at the heterozygotes, there's quite a significant impact on liver function in people carrying 1 copy of this splice variant. So we see a significant elevation in some of the liver enzyme levels, and we see a 3-fold increase in liver disease, non-viral related liver disease in people who are carriers versus not. Interestingly, for folks who are familiar with liver disease genetics, there is a very well-known common variant in a gene called PNLA3, which underlies NASH and NAFLD and is especially common in Hispanic Latino populations. We showed no association with that particular risk factor. So we think we found a second risk factor for liver disease in at least Puerto Ricans. Go ahead, Lisa. Yeah. That's such a great idea. So we thought about that, but we didn't have big numbers. We only had 43 cases in this case. So we didn't really have big numbers to see that. And our biobank skews older, so it skews people in their late 50s, early 60s. And we don't have the historical records going back for pregnancy sometimes. But I think that would be a great thing to look at. We did not see any significant skew in terms of women being more affected than men. But I think it would be really nice to go back and look at what are the modifiers for these particular bad outcomes that are driven by this variant. Great question. But the other point I want to make, and this is back to how important biobanks and diversity in genomic resources can really be, is when we go back and look where in the world is this variant, we find that it is common in Puerto Rican populations, so 2% of the population have it. So that means 1 in 50 are at risk for these heterozygous effects that we're able to uncover, although of course these would need to be replicated in bigger studies, and we're pursuing that. And then 1 in 5,000 at risk for these much more severe outcomes of carrying 2 copies. And it's not just an issue in Puerto Rico. There's 8 million descendants of Puerto Ricans living everywhere in the world. So people walking into your health system could have this disorder as well. And this is the kind of ways where we can start to leverage these resources to start to make clinically relevant findings that can go back into the clinical knowledge bases that are used in doctors' offices. Our study didn't bring it quite there yet. What we would like to do next is actually set this up as a prospective clinical trial and start to look at younger people and really describe clinically better what's actually going on as a basis to generate evidence to develop clinical guidelines for people coming into the clinic and testing. Where we can start to do that already, and again, this is where biobanks come to the rescue again, biobank propaganda hashtag, is where we already have evidence that seems to be aligning, that can start to be brought into medical programs or new medical programs, and how biobanks can help you do that. So back in 2018, we were very interested in turning our biobank from a research engine into a research and clinical engine. And we were interested to do this for genomic screening. So genomic screening today are mainly monogenic risk variants that you can have or harbor, where if we know about it, we can either act to prevent disease in people who have symptoms. So knowledge of that variant and the disease it's associated to, we do has medical actionability, that is either health management or intervention and therapeutics. And that they're highly penetrant, so you're very likely to develop this disease if you're harboring those variants. So today, there are a number of federal and medical bodies that issue guidelines around specific variants and disease associations. One of them is the CDC at Tier 1. That happens to be where we started in 2018. I should say this work was driven by Nora Abul-Hassan. And we started identifying individuals who had pathogenic variants in any of these 10 genes linked to these 3 diseases and contacting them, asking them would they like to receive these results. When they did, we would bring them back in, we would counsel them on these findings and refer them appropriately to follow-up care. And that actually took a good bit in terms of infrastructure. It sounds very simple, it wasn't. It took a lot of infrastructure to get that off the ground, but we did. But one of the things we were really learning at the same time on the research side, that there was mounting evidence that a gene that was not yet on that list was under consideration by ACMG, but not yet on any of the lists that you would use to return findings. Was a TTR linked to a hereditary form of cardiac amyloidosis. The reason that came to our attention was twofold. Number one, that there's a particular founder mutation at 4% in African American populations, and this is an autosomal dominant disease and highly penetrant variant. So we knew that there was a sizable population at risk for this inherited disorder. And number two, in 2019, 2 new therapeutics came on the market. Previously, there had been none that could actually start to treat symptoms of this disease. So now the clinical evidence is moving this into a medically actionable bucket, and the population prevalence evidence is saying that this is affecting a large population. So we felt it was imperative to start to build the evidence, to build out a clinical program around that. So we first assembled some of the evidence. Having generated the evidence, we assembled a committee of clinical experts. We presented the evidence. They ultimately agreed with us, and then we started returning results for TTR in Mount Sinai, so that today, if you're in the biobank and you harbor one of these variants and you want to receive genetic findings, we will be able to inform you of that risk, and then we have a high-risk clinic that patients are referred to. The other thing, and we're getting to 1:05, so I'll try and hurry up. The other thing to think about as we're starting to think about how to bring genetic information into clinical care is what people are going to think about. And people we usually think about as either patients or providers, medical professionals, but you can be broader than that. You can think about what health systems are going to think about it, what communities and society is going to think about it. But certainly we ought to ask, because it can be very valuable to understand outside of how researchers think, what other people think about this information that's going to be so, we think, helpful, but maybe others will disagree. If you ask diverse communities in New York City about, this is a study of about 7,500 people, a vast majority, 93% of people would opt to receive this type of information about themselves. Interestingly, I'm not showing it here, but it's in the paper. If you break it down, certain communities have higher rates of interest in this information. I find it very interesting that those communities tend to skew younger. Maybe that's not so unsurprising. Tend to be women over men. Again, maybe people would guess that. Tend to be Hispanic Latino over other race ethnicity groups. So people's attitudes can actually surprise you. So I'm going to finish up with polygenic risk. I feel I can speed through a little bit of this because Iran has done my job in introducing this. I'm going to assume you all know what polygenic risks are and the evolving conversation, in some part kicked off by this paper in 2018, but actually we've been doing decades of work on this. I think the real tipping point has been that our genomic databases have grown to the point where we reach the tipping point of prediction accuracy, where we can now look at deciles and tails of polygenic risks that have similar risk magnitude to the types of monogenic risks we were just talking about. So in this case, 8% of the tail of cardiovascular risk in this example have the same 3 to 5-fold increased risk for cardiovascular disease than people harboring a rare variant at one of the familial hypercholesterolemia genes. Of course, we also know by work of many groups, including my group, including many groups here, and this is work from Alicia Martin's group, that those polygenic risk predictors perform less accurately when you move them out of populations that are close proxies for training data that goes into those models, and you can see that the further divergent populations are to the populations that were used to develop the models, the worse the predictors do. We started observing that in New York, that this wasn't just true if you start to think about moving these predictors out of European populations into non-European populations. What happened? Gosh, who's punishing me? Again? No? Dead battery. It's not my fault. Okay, can I just shout a bit and then, okay, and I'm almost finished. This is also true if you start to look at much more fine-scale populations. So, for example, in the BioMe Biobank, 98% of participants who self-report as white or European ancestry fall into two communities. One is highly correlated with people who also say they're Ashkenazi Jewish, and one is highly correlated with people who just say they're European ancestry. About 40% in Ashkenazi Jewish, 40% non-Ashkenazi Jewish European ancestry, and which is fairly typical of the demographics in New York. If you look at a polygenic risk predictor, in this case for type 2 diabetes, and you normalize it and you do all those nice things, and then you just stratify your population by whether or not you're Ashkenazi Jewish or non-Ashkenazi Jewish in New York City, you see a mean shift in the distribution of that score. That has just got to do with whether or not you're in one of those two groups. And this is a significant mean shift. Does this mean shift mean that the predictor will perform differently? The answer is complex. So for many diseases, and here we looked at 5, we ultimately looked at 18. For many diseases, we see that there is some difficulty, some noise, some toggling, but it's not really significant, so maybe at the end of the day it doesn't matter. And for some diseases, we see it absolutely matters. If you're starting to look at the deciles of the distribution, you clearly see that this predictor, for example, for IBD, a different IBD, irritable bowel disease, is much better predictor in Ashkenazi populations, much poorer predictor in non-Ashkenazi Jewish populations. The reasons for that, we can guess, there are more Ashkenazi Jewish populations in IBD research, and that might be informing the training data, which is informing the tool itself. You could also discuss the architecture of the disease, the ways in which you're constructing the PRS, lots of complex reasons. But I'm pointing this out. I will also point out work from other groups, and we've done this as well, where we start to look at nuances of fine-scale structure in the UK Biobank, even in people who self-report as White Britain in the UK Biobank. Historically, we think these are a very homogeneous group. No, there's a lot of fine-scale structure. This fine-scale structure really impacts these predictors that we're using if we're using them naively, which is what I want to come to at the end, because I really think that the way we solve at least part of this problem, of course, is empirically by bringing more diverse data into these predictors. We're still in a stage here in 2022, 96% of our data is going to be European. So what do? We're not going to solve that problem in the morning. Optimization and calibration are very powerful things that we can do. Methods that actually model demography into the scoring are powerful things that we can do. Appropriately dealing with population structure and stratification are very powerful things that we do, and there's a lot of gain to be had there. It is not going to solve the whole problem. It certainly can improve the types of scores that we build. And part of the reason is that human history really is super complex, and it is very tied to what we usually term as E, which can be social, political, economic indicators of health. So those are things we often don't directly model, except for Iran is showing how we can do that with some methylation marks. Others are starting to think about how we can do that with social determinants of health brought into these models, or environmental indicators of health brought into these models. Either way, we're going to have to find a way to integrate the genomic piece that tells us about the inherited risk and actually correlates and maybe is a biomarker for some of the non-inherited risk into how we think about risk for disease, which today includes clinical risk factors, not often actually, but sometimes also family health history, and how we actually bring all of that information into predicting risk. Again, bringing this back to what people outside of our scientific community think about it often. Their attitudes are very open. Here's a study recently that we did that was a qualitative study of 30 Spanish and English speaking diverse participants in research in New York City. And the interest and perceived utility of polygenic risk information was very high, even after explaining how poor these tools or how less accurate these tools are in non-European populations. What was a bigger concern were access issues, transportation to the doctor, and whether or not an individual could be empowered to do anything with risk information. And that's going to stay a problem even when we start to build better risk prediction tools. So I'm going to end with a phrase friend and colleague Vince Bonham brought up in a talk he gave a couple of months ago, and it just struck me so much. And I asked him, I said, hey, I really want to borrow that for talks because I think this is so important. And he brought up this point, which I think just gets to the kernel of the issue, that bringing genomic information increasingly into clinical environments or public health environments or wherever else we want to think about this is likely going to be a small slice of the pie in terms of the full picture of health. So we can improve those tools, we can get them to do better, and we can get much more accurate at predicting that slice of the pie. But likely that's not going to ameliorate current health disparities, because we know structural health disparities have a much bigger impact on health, on human health. However, if we fail to address the lack of diversity and the problems that is bringing to the tools that we're building from these resources, then that will very likely contribute to health disparity. And that's what we have to do in the next 10 years. And actually, when I say we, I'm really looking at all of you. with that, I'm just going to spend 2 minutes thanking everybody who did the work. You heard a lot of work from Gillian and also Ruhi. Oh, I'm embarrassed. Sinead Kulina is around here, and she did some of this work as well. And others in the Institute, Sabrina Sukiel, Jackie Otis, and Emily Soper. Longtime collaborators, Chris, Alicia, Carlos, and Jen. And obviously, thanks to all the thousands of people who have volunteered for research in the BioMe Biobank. And like everybody else, I will point out that New York is fun and fantastic. And if you're interested in this work, please reach out to me. Always looking for both faculty and postdocs. Thanks very much.